used to look like when we get them. And literally, this thing was at least this long. Oh, is that you making the noise? Okay, this thing was at least this long, at least almost 2.5 feet. So as opposed to what, 13 inches? Is that what we've been dealing with? All right, and here you can clearly see. Look how obvious the external gill slits are on this. And then there's the pineal eye, and right in front of it, you can see there's a little hole right there. That's the median nostril. All right, there's the lateral. Can you see the lateral eye right there? So now, if I ask questions on the test that are like this, you'd get them, right? Isn't this a lot easier than looking at their actual specimens? So I want to make this reasonable for you, to the best of my ability. You're still going to have to study, okay? All right, so this is what you'd see on the test. You might see a diagram that looks like this. This is a drawing out of your book, and what is it? Acorn worm, okay? And then what would this be coming down along here, this dark line? The what? The ventral nerve cord, right. And therefore this up here would be the what? And notice how there's no real cavities in it, but you see the hint of a cavity there and there. Because again, these don't necessarily, they're not true chordates, right? These are hemichordates. And then so what is this structure here in the drawing called? That's the stoma cord. And then what's this little thing right here? Glomerulus. And what's this space? So I will probably use the actual pictures of the specimens to the best of my ability. But if there's something I want to put on the test that I just can't see, I haven't seen a ventral nerve cord on any of them that I could ask. When we used to just do the slides, occasionally I could make one out. But even then it was kind of suspect because they weren't clearly identified. So spend a little bit of time with this picture. There's a possibility it could show up. I'm not saying it will, but you should know the major structures. So when you're studying it for the lab test, you know, and then don't be afraid to read the descriptions of structures. It will help make you understand them better. This is what the actual the actual photomicrograph of the specimens look like. This is the glomerulus right here, see? And this is the proboscis alone. So this is just part of the tissue of the proboscis, right? Okay, there's your stoma cord right there. See it? Would anybody miss stoma cord, or is that fairly obvious? So if I put that on the test, you won't be upset, right? What are these? Tongue bars, right? And then the spaces between them. So these are actually little U-shaped things that are tongue bars. What are the spaces? Those are the internal gill slits. Okay. The tissue here, the tongue bars. See how it comes down. In the drawing, they look like little U's, right? Go back to drawing. See how it looks like a little U? See, come down, come down, come down, right? And then when you actually get into the what they actually look like, see, comes down like this, comes down like this. See how it comes, makes a little curve, and then the slits are just the spaces in the middle, in between them. Okay. This is an adult what? Tunicate. So make sure you know the incurrent siphon and the excurrent siphon. All right. Here's the digestive tract coming down through here. This is all digestive tract, the intestines. And then what's this little opening here? Anus. That's the anus. What's this little thing right here?
The nerve ganglion.
The nerve ganglion, right? There's a little nerve ganglion. And then what are these little things coming down through here? Again, what kind of feeding style does this have?
Filter.
So you're going to have to pull in water and filter through slits, right? And then they collect into a thing called the atrium. I don't know if they have atrium listed in the book, but and then the endostyle is right down here along the bottom. I'm not sure. I'd have to look at the drawing. I haven't looked at it, but the endostyle is that sticky mucus covering channel that collects the particles, the food particles, just like the amphioxus has it. In the amphioxus cross sections, you can see an endostyle clearly. It's right at the very bottom. It's that little U-shaped evagination, if you want to call it that. But anyway, so go through and make sure you look at these structures and get to know them, because there's this and there's this, another view. There should be two views from each side. So, but this is there's a possibility that one of these drawings can show up on the test. Okay, and then this is an actual photomicrograph of the slide that we call amphioxus, branchiostoma, or cephalocordate, or lanceolate. All those 4 names apply to it, right? Why is it called a cephalocordate? Because the notochord, which is this thing right here, right, comes all the way out to the tip of the snout, right? It actually extends out here. And then the nerve cord, which is a dorsal hollow nervous system right up here, it's above it, right, has these little pigments, has these little pigmented ocelli, O-C-E-L-L-I. They just call them, what do they call them? Photoreceptors. Yeah, these are photoreceptors, except for the one at the end, the little dark one right on the tip. That's just simply referred to as the pigment spot. And then sitting above it, this isn't as good. I wouldn't use this on the test because that's not as good as what I'd want. But if you know there's a pigment spot here, what's this little dark structure right there? Right. I wouldn't use that on the test because I'd have a better picture for that. Okay? But I'm just saying, this is how you have to know it. You have to look at it at this low mag and then go up to the high mag to see the details. What's this whole thing right here called, from here to here? The what?
Wheel.
Right. And what's the top one called? Groove. And then see the little indentation right there? See how it comes up and make a little dip right there? What's that? Hatchek's pit. Right. What are these? Oral cirri. Right. And then this is the velum right here. See this thing coming down here? That's the velum. And I'm wiggling it on purpose. Because coming off of it, there's one coming out, this one's coming out like this towards you, probably out like this towards you. Kind of see thing there. There's little things come off called velar tentacles. And again, I might have a slide that has those really clear. This isn't all my slides. I've got more slides to go through. I mean, I gave you a pretty comprehensive set of slides, but I might have some that are even clearer, so that I save for test purposes. This is your peripharyngeal band right along here, right? And then right across the top, what's this going to be called coming up here since it's above? What's this going to be? It's above, so use the word epi. It's the epipharyngeal groove. Right. Is any of this familiar? Have you been studying this? And it is a lab test on Tuesday for those of you who aren't sure whether it's a lab or lecture. I'll just make that clear. I don't want any confusion. All right, this is another view. Now in this view, again, see the notochord going all the way out to the tip of the snout? Okay, here's the nerve cord. All right. Let's see, here's the wheel organ right here. And here's the oral cirri. This is what we had with the old technology. That's what I had to use. This is, I told you about the little arrow generator thing that we had, so that was kind of cool. They actually, the reason I put it in here, because see these little dots? There's actually little structures in there that make that they don't make it hard, but they give it some support. So if you actually look at them under very high mag, you can see these little, I guess they're calcifications or something that are in there. But and this is the drawing. When it comes down to it, you might see this too. I told you, you have to be aware of what's labeled in the drawings. Did I not make that comment earlier? I don't like to use these, and I will use them sparingly, but there's probably about a 75 to 80% chance you'll see a couple of these in the test. I'm not going to say 100% because I don't want you to, then if I don't put one on, you said you put drawings on there. Okay. All right, and this, again, this shows you the wheel organ, which Hatchek's groove, Hatchek's pit. There's your pigment spot. There's your flagellated pit. Okay, here's your notochord. Notice they do it like this with the little lines coming down because if you look at it under high mag, you actually see those little lines. Here's the velum coming down here, and these are the drawings of the what? Velar tentacles. Okay? So, again, take a few minutes and look at this. I would say your best thing to do would be sometime today or tonight, or even after we get done in here, just go look at these pictures and look at the slides that you have on your computers and then get something in your brain. And remember what I told you the other day. Go down that list, and if you can visually imagine the pigment spot, or the flagellated pit, or the notochord. Then you're done with those. You got those down. You've learned them. Just focus on the ones you don't know. So I think you're all going to kick butt on this lab test. That's my prediction. Don't let me down. Okay, that's a test quality flagellated pit. And again, if you missed that one, remember histology. Okay. Here's your notochord, again, your Siri. So I'm limited to what I could ask on this because it's just the front tip of the snout. So, but you have to be able to go low mag to high mag, you know, because you're going to see things at both levels. The thing in histology is that a lot of students didn't spend enough time. Well, many did, but a few students didn't spend enough time going from low mag to high mag. Like if you look at the liver, a very low mag, you can see the central vein and all the sinusoids converging on that central vein. If you look at a very high mag, all you see are 2 liver cells with canaliculi in the middle. They look totally different, but you have to know it's still liver. So that's the thing. That's why you can't look at it and say, okay, I know what that is. A lot of times people do that in different types of labs. As soon as they see one specimen and they think they got it down, but then not everything's alike. Yeah, a lot of these things, remember the differences in the amphioxus that you've seen just in a few minutes we've been doing this. This shows you, remember those lines in the notochord I was telling you about? See how the little, because it's actually a series of little discs of tissue, you know, going back, but you can clearly see the vertical lines. So that's why I like the way they drew that one. And again, notice the flagellated pit right at the top, and then there's the pigment spot. I've seen more impressive pigment spots in my life, but that'll get through. I think this one would be used for these right here. That's probably what I would use, or I could do the notochord, you know. So. Now, enough said. But if I would have put this on the first day and said, on the pretest, said, okay, what's this? I didn't do a pretest in here, did I? But this is what I would have been on. I would have thrown this up on the pretest, and you all would have looked at me like, "Yeah, you think we're supposed to know what the hell that is?" Okay. But it's not that hard now, is it? If you actually look at it and think about it, this is the way you acquire knowledge. You just kind of sit and work with it. So, will you use this again in the future? I don't know, and I don't care. But you're going to learn it now. So it's all the ability to learn how to assimilate information and process it, and hopefully retain it. High mag of the wheel organ, okay? Again, first day you'd be going, what the hell? You know. So. I don't know why, but I look at it. It looks like little lightning bolts going through here. I don't know. Maybe I'm just weird. Electrical charge, doesn't it? I could see turning that 90 degrees and making it into a painting on your wall. So. Anyway. But that's the wheel organ, you know. People walking out there looking and going, what the hell is that? All right, now we're getting to the area of the velum, okay? This is the velum coming down here, and then here is the peripharyngeal band coming in at an angle. And these are your gill bars, and these are your gill slits in between. I was trying to get velar tentacles, but it's too blurry. I can't really make them out. Again, if I can't get good, solid, clear-cut pictures for the test, I will use the drawings. That's when the drawings come in. Okay? All right, we're further back, right? So what's this part called coming down through here? What's this in this region? It's the esophagus up here, right? But what's it called when it comes back here? Here's the ileocolic ring right here, right? Mm-hmm. They call this the stomach.
They have esophagus, hepatic cecum.
That's this right here.
Yeah.
Yeah. Okay. In some lab books, they have referred to this. See how it's kind of expanded right here? Mm-hmm. Again, your book might not have done it, but some books do call that a stomach. But it's questionable. That's why I guess your book didn't put it in there. I have another functional anatomy of the vertebrate books that does call that the stomach. But this is called the hepatic cecum, or the midgut cecum, or the hepatic diverticulum. Which is it called, cecum or diverticulum?
Cecum.
Okay. In your textbook, I think it calls it a diverticulum, doesn't it?